Okay, I wanted to quickly talk about the evidence for evolution. So, the three main observations that help support evolution are homology, biogeography, and the fossil record. This illustration here actually shows some evidence through homology. So, if you look, all four of these organisms have a opposable thumb that's used for grasping, like grasping trees. If you look at the chimpanzee, though, and the human and the gorilla, you'll notice that they're made up of three fragments of bones. Those bones are developed from actually the same embryological tissue and show that the chimpanzee, the human, and the gorilla share a common ancestor that originally developed this opposable thumb. If you look at the pandas, their thumb actually develops from this bony fragment of the wrist. And it's very different in structure from the chimpanzee, human, and gorillas, showing that the panda does not share a recent common ancestor with the three great apes. So homology is a similarity that you see that shows common ancestry. And there are three main types of homology we're going to discuss: anatomical, vestigial, and molecular. So anatomical homologies are either bony structures or other structures that develop from the same embryological tissue and show that the organisms share a common ancestor. They might have a different function, but they actually develop from the same structures. So if you look at all the highlighted bones here, the purple ones, the red bones, those all develop from from the same embryological structures. However, they have different functions in these organisms. Like all the bones in the human are very closely related to the bones in the cat, but the human arm is made to be very agile and have a lot of dexterity. Versus the cat, their arms are mostly meant to propel them forward. And in the whales, they're used to help the whale swim. And in the bats, they have these huge long digits that help provide enough surface area for lift for flight. All four of these organisms share a common ancestor, and we could see that with these homologous structures. They're all mammals, but they all use those bones for different functions. Studying also how embryos develop can allow us to see homologous structures that we can't see in the adult organism. This study is known as comparative embryology. And when we look at the embryos, we can see some structures that they don't have as adults. For instance, all chordates and vertebrates have pharyngeal pouches and postanal tails. Now, as adult organisms, humans don't have a postanal tail, but during embryological development, we do have a postanal tail. We also have these pharyngeal pouches. Pharyngeal pouches in humans become part of our head and neck. In other organisms, they become part of their gills in fish. Vestigial structures are structures that are basically homologous structures, but no longer have the same function as what was found in the common ancestor. So the appendix is the most common example of vestigial structures. If you take a look at this illustration down here, what the appendix is it's an extension off of the cecum. So this shows the small intestine, the last section of the small intestine called the ileum, and here is where it meets up with the colon. And off the end of the colon is the cecum. And in humans, there is an extension of the cecum that we call the appendix. Now, our common ancestors had a longer cecum that had a huge amount of space for bacteria to help digest cellulose. However, in humans, it's much smaller, that same section, so that's why it's called the appendix. It's no longer big enough for bacteria to be housed to digest cellulose. It does actually have some immune system function. Wings on an ostrich are considered vestigial structures as well because they no longer help the ostrich fly. There's a tailbone in humans that's considered a vestigial structure because it no longer has the rest of the tail attached to it. And leg bones on a snake or whale are also considered vestigial structures. So there are some like bony structures in a snake and a whale, but no legs attached to them. This shows that they shared a common ancestor with tetrapodal organisms, four-limbed organisms. So for instance, in this image of a whale here, there are some pelvic bone structures in the rear of the animal here. And these pelvic bone structures show remnants, really, of a tetrapodal organism of hind limbs. Whales, after all, evolved from terrestrial mammals. An exaptation is a structure that evolves in one context but is co-opted for another function. Sometimes people confuse this with vestigial structures. An exaptation, though, is a structure that adds really a new function to the original structure. So feathers originally evolved in birds, not for flight, but actually for keeping warmth. So we see actually feathers and also other non-flying dinosaurs that went extinct. So what's the difference between an exaptation and a vestigial structure? Because actually some structures can be described as both, it can be confusing. But whenever we talk about a structure having lost the original function, that describes a vestigial structure. An exaptation is something that we're talking about where a structure has gained a new function. Vestigial structures provide evidence for common ancestry. They show that they're homologous structures. Exaptations show that there has been a divergence from that original common ancestor. So, like I said, some characteristics can be described as both vestigial and exaptations. Penguins' wings, for instance, may be described as vestigial structures because they no longer function in flight. But the wings, since they also help them swim, could also be described as an exaptation. Also, the appendix could be described as vestigial because it no longer has the function to house bacteria to digest cellulose, but it does have some immune system function, so that could be described as an exaptation. The third type of homology, which is actually the main homology that we use now to show relationships between organisms, is molecular homology. So that means looking at genes or proteins or other molecules that show similarity and differences between organisms. This table over here, or graph actually, shows the similarity of amino acids in part of the hemoglobin, one of the hemoglobin polypeptides. Humans, compared to rhesus monkeys, have 95% similarity in amino acid sequence between the hemoglobin found in both the organisms. Remember, hemoglobin helps us carry oxygen, so it's pretty highly conserved protein. It's an important protein. Humans, compared to the mouse, though, only have 87%. That shows that we have a more distant common ancestor with the mouse than we share with the rhesus monkey. Rhesus monkey, again, we share more similar amino acids. And then if we look, further down, chickens, obviously we share a more distant common ancestor. We have a more different hemoglobin sequence. Frogs even more distant, and lampreys even the most distant. So some molecules that we can look at, of course, are DNA from the nucleus, or we can look at DNA from the mitochondria or the chloroplast. Looking at those different molecules can provide different sorts of evidence that we might be looking into. Nuclear DNA, for instance, recombines a lot, so sometimes it's a little challenging to try to trace when changes occurred. Mitochondrial and chloroplast, on the other hand, are passed down maternally, and so you can track. There's no recombination actually occurring, sexual recombination occurring in mitochondrial and chloroplast DNA. So it's easier to track back when changes occurred. Also, mitochondrial and chloroplast DNA mutate more rapidly, so it's easy to use them to figure out differences between closely related organisms. By the way, mitochondrial DNA is referred to as mtDNA, chloroplast is referred to as cpDNA. We can also use mRNA, we can use rRNA. Ribosomal RNA or rRNA is actually really great for looking at organisms that are more distantly related. Ribosomal RNA is highly conserved. Protein synthesis is very important for organisms to survive, and so we see only big changes occurring between very distantly related organisms like humans and bacteria. Okay, the next piece of evidence that we use for evolution is biogeography. Biogeography means that we study organisms and where they live, and we look at how they're related to each other. Now, generally, when we look at organisms that live close to each other, they're more likely to be related to each other, and they're more likely to share similar sequences, for instance, homologous sequences. Organisms that are endemic, only found in one place, are going to be similar to organisms found somewhere nearby. For instance, the marine iguanas that live in the Galapagos, they're similar to the terrestrial iguanas that live in South America. One interesting thing about studying organisms and where they live is actually similar pressures on organisms can actually cause similar evolution to organisms that are even distantly related. So when we have organisms kind of evolve structures that have similar functions but don't come from the same genetic material or the same embryological tissue, then we refer to that as convergent evolution. So, for instance, these three wing structures here in the insect, the bird, and the bat are all considered analogous structures. Bats, birds, and insects do not share a common ancestor that had wings. They all developed those wings separately from organisms that did not have wings beforehand. You can see that the insect wing is very different in structure from the bird and the bat wing. Now, one thing that is a little bit confusing is if you look at the bat and the bird wing, there is some homologous structures. Bats and birds do share a common ancestor that was a quadruped that had four limbs, right? And that, was terrestrial. So they do share similar homologous structures. That's what the different colored bones are representing there. Those are showing the homologous structures. However, if you look at the wing structure, they did not evolve from the same structures. Like in the bat, they're from the digits really extending. And then if you look at the bird, there's really this large structure here, like wrist bone basically structure, that extends. And the feathers are what actually bring a lot of flight. Another example of convergent evolution is between these two flying little mammals. The flying squirrel that is native actually to southern North America where we live, and the sugar glider that is native to Australia. Both of these organisms fly from tree to tree or glide from tree to tree using this flap of skin that they have. However, the flying squirrel that's from North America is a eutherian mammal. It has a well-developed placenta. Sugar glider, on the other hand, has a undeveloped placenta, and the sugar glider, like all other marsupials, they're born at a really young development age and then kept in the pouch by their mother until they develop more. So we have two different types of mammals that developed both the same structure. So this is convergent evolution. And these structures are considered analogous because they don't come from the same really genetic material or embryological tissue. Parallel evolution is where we have organisms that share a common ancestor, have undergone similar pressures, have been exposed to similar pressures, and have ended up evolving similar characteristics. So as an example, there are a couple species of cichlid, really, that have evolved really from one singular species of cichlid in Africa. And in these different lakes, they've actually undergone similar pressures. And in each of the lakes, there has been divergence among species to produce characteristics that are the same in each of the lakes. So if you look at these two lakes here, Lake Malawi, and I'm going to say this wrong, but Lake Tanganyika. Tanganyika is what my daughter says. In each of these lakes, you'll see fish that have same coloration patterns. But they actually share more recent common ancestor with the fish in one lake than they do to each other. So here's a quick diagram that shows the difference in these evolution patterns. Usually when we think about evolution, we think of divergent evolution, organisms that have a common ancestor that diverge. And homologous structures show that organisms share a common ancestor. In diagram B, this shows the idea of convergent evolution, where you have organisms that have a distant relative that are put underneath similar environmental pressures and develop structures that have similar function. And in diagram C, this shows parallel evolution, where we have organisms that are under similar environmental pressures and evolve similar characteristics, even though they may share a more recent common ancestor. The third piece of evidence that we look at is the fossil record. The fossil record helps provide us really patches to show how organisms share a common ancestor. Fossil record, we can find transitional species. Transitional species are species that show the relationship between two organisms. So, for instance, whales are related to terrestrial mammals, and there are fossils, transitional fossils, that show that there were these aquatic mammals that had still remnants of hind limbs that show they're related to other terrestrial mammals. We'll talk about this question tomorrow. That's all, guys. Have a great day.